Introducing the VitroView TM COX SDH Double Histochemistry Stain Kit SKU VB3022. This reliable tool is essential for detecting mitochondrial abnormalities in frozen tissue sections. VitroView Biotech is committed to making tissues, cells, genes, and molecules visible. For more information, visit our website or contact us directly. Understanding COX SDH and mitochondrial dysfunction is crucial. Cytochrome C oxidase, COX, is the final enzyme in the mitochondrial ETC, producing ATP. Mutations can impair COX activity, leading to dysfunction. Succinate dehydrogenase, SDH, links the Krebs cycle to the ETC and remains unaffected by mitochondrial DNA mutations, serving as a marker for COX-deficient cells. The COX SDH double stain is a dual enzyme histochemical technique used on frozen tissue to identify mitochondrial dysfunction. It detects cells with deficient COX activity and preserved SDH activity, visualizing mitochondrial respiratory chain defects at the cellular level. This method is essential for diagnosing mitochondrial diseases and studying metabolic defects. Research applications of the COX SDH double stain are vast. In aging research, it reveals mitochondrial mosaicism and tracks COX loss. In muscle biopsies, it diagnoses mitochondrial myopathies. It also aids in studies of neurodegenerative diseases, cardiac pathology, animal models, and cancer research, providing insights into COX SDH imbalances. The VitroView TM COX SDH Double Histochemistry Stain Kit provides reagents for staining 50 to 100 slides. It includes COX A and B solutions for Cytochrome C oxidase detection, succinate solution, and SDH incubation medium for succinate dehydrogenase activity, and enzyme-specific inhibitors for control validation. The kit is optimized for frozen tissue sections only, ensuring the preservation of mitochondrial enzyme activity. All components should be stored at -20 degrees to maintain stability and staining performance. Let's briefly go through our in-lab COX SDH double staining process. Cryo sections are cut at 10 to 14 mm and thawed onto slides. To begin COX staining, thaw and mix COX A and B solutions. Rinse slides in PBS to remove OCT, then apply 80 to 200 ml of COX solution. Make sure tissues are completely covered by the solution within the hydrophobic pen-defined area. Incubate slides at room temperature in the dark for 1 to 1.5 hours. Check staining visually, then rinse slides again in PBS. Next, prepare SDH incubation medium by mixing succinate and yellow SDH solution at a 1.5 ratio. Apply SDH solution to the slides and incubate at 37 degrees for 40 minutes. Slides are then dehydrated in 95% and 100% ethanol, followed by 3 xylene washes. Finally, apply mounting medium and cover slip the slides for imaging. Proper tissue preparation for cryosectioning is critical. Collect tissues rapidly without fixation and freeze on dry ice. Preserve by freezing in isopentane or propane chilled with liquid nitrogen, then store at 80 degrees. For sectioning, embed frozen tissues, cut 10-14 cryostat sections, and store slides at 70 degrees until use. The COX staining protocol involves preparing the COX incubation solution, rinsing slides, and incubating with COX solution. Apply 80-200 UL of COX solution onto slides and incubate in the dark at room temperature for 1 to 1.5 hours. Check staining and extend incubation if needed, then rinse slides in PBS. For SDH staining, prepare the SDH incubation solution by mixing succinate solution with SDH medium. Incubate slides with 80 to 200 UL of the prepared medium at 37 degrees C for 40 minutes. Dehydrate with ethanol, clear with xylene, and mount with a suitable organic medium. This process ensures precise visualization of mitochondrial dysfunction. Specificity controls for COX SDH activity are essential. For COX, apply the inhibitor solution before step 3 of the staining procedure. For SDH, replace step 7 with the inhibitor solution. These controls confirm the specificity of the staining process, ensuring accurate identification of mitochondrial dysfunction. Expected staining results show normal cells with brown staining, indicating active COX and SDH. COX-deficient cells appear blue, showing COX dysfunction. Specificity controls confirm COX and SDH inactivation. This staining method clearly visualizes intra-fiber mosaicism, aiding in the study of mitochondrial abnormalities. The VitroView TM COX SDH double histochemistry stain kit is featured in several peer-reviewed publications. These studies highlight its application in analyzing COX activity in various tissues and its role in understanding mitochondrial dysfunction. The kit's reliability and precision make it a valuable tool in scientific research. Ordering the VitroView TM COX SDH double histochemistry stain kit is simple. Visit our website, email, fax, or call us to place your order. At Vitro Vivo Biotech, we are dedicated to providing high-quality products and exceptional customer service. Contact us today to enhance your research capabilities.